Hello everyone, my name is Geremy Clair and I'm from the Pacific Northwest National Laboratory. Today, I will present to you the tool Lipid Mini-on. This R-based tool is made for automatic lipid ontology generation and enrichment analysis. Full disclosure, today I will talk to you a lot about the lung, as my group is part of the lung map program. But I promise, I won't talk about any coronavirus. The lung map program is an NHLBI founded program. Its goal is to generate a multi-omic atlas of the lung during its normal development, especially after birth. The schematic shown here is showing you the different analyses that we are performing at PNNL, and I will not spend time on details here. So, why should we care about what's happening inside the lungs after birth? Thanks to medical advances, and in particular to the development of artificial lung surfactant in the past 30 years, the number of children surviving premature birth has grown tremendously. The CDC estimation is that nowadays, 1 out of 10 children in US is born premature, and this can lead to abnormal development, to overrepresentation of some cell types, to problems in surfactant production, leading to a variety of diseases, including asthma, bronchopulmonary dysplasia, pulmonary hypertension, fibrosis, and so on. As you can see on this confocal microscopy image, the lung is composed of various cell types and multiple structures that participate in the respiration and in the defense against pathogens. The development of these fine and elegant structures is occurring both prior and after birth. The early exposure to air impacts tremendously the developmental program of the lung. Because each individual cell type is contributing in a different manner to the biology of the lung, it's really important to know what molecules are specific to each one of them. To explore this, we dissociated the lung into a cell suspension. Then, we sorted by fluorescence activated cell sorting the four main populations composing this organ: the endothelial cells, epithelial cells, mesenchymal cells, and a mixed immune cell population. When sorted, we used a modified Foch extraction called MPLEX to extract from the exact same sample polar metabolites, proteins, lipids. For more details on this method, I recommend the paper by Nakayasu and colleagues published in 2016 in M-System. The lipids were then analyzed by LC-MS on an Orbitrap instrument, both in positive and negative mode, to ensure a good coverage of diverse lipid classes. Then, we analyzed the data generated during this campaign using our now standard lipidomics processing pipeline. The different lipids present in the samples were identified using an in-house developed software called Liquid that enables manual inspection of the spectra, leading to identification. Then, we used the very good MZMine software from Tomasz Puska and colleagues to align the chromatographs and perform quantification based on area under the peaks. Finally, we used in-house made R packages for multi-omics analysis to perform data filtering, normalization, group analysis, and statistics. This is where the trouble started for me. I had a very nice, rich lipidomic dataset, and because my knowledge in lipids was rather limited, I called my favorite lipid expert, Jennifer Kyle. Hi, Jennifer. I've done the stats on my lipidomic dataset. What should I do next? Usually, I take the lipid.csv file, I open it in Excel, I extract the significant ones. Then, I group the lipids by category, main class, subclass. Check if there are any trends. Next, I sort them by the total number of carbons and total number of double bonds. Then, I looked at the individual fatty acid composing the lipids, and so on, and so on. So we ended up with a very nice heat map, but I decided I didn't want to go to that kind of struggle anymore. At about the same time, I was working on a proteomics dataset describing the temporal modulation of the protein of the human developing lung. And very quickly, by doing a simple online enrichment analysis with David, I was able to figure out what pathways and ontology terms were enriched in the different clusters, and to figure out that there was a global reorganization of the matrix during postnatal development, results that we've validated since then. Unfortunately, at the time, there was no available tool to perform the same type of analysis on lipids. One way to make fat and lipids is to eat sugary, right? So I will take the example of a delicious Haribo Gold Beer bag to introduce to those of you who are not familiar with the concept what an enrichment analysis is. So, I have a Haribo bag and I give 5 to my little sister Alize, 4 green and 1 orange. And of course she complains. The green ones are disgusting. The question is, did I give her purposely a set that was enriched in green Haribos? To know that, we need to look at what's left in the bag. And we can see here that there is a lot of red, yellow, and white delicious ones, but only one green one left. We can use some statistics, for example here a Fisher exact function, to calculate the probability that I gave her purposely a set that was enriched in green ones. So we make a contingency table and calculate that probability. And the probability that I didn't give her that purposely is 1 out of 1,000. So that means that it's very likely that indeed I'm an evil brother. One thing that is important to remember is that we need categories to be able to do enrichment analysis. And those categories need to be available. This is why I started to look around in databases to try to find satisfactory association between lipid names and ontology terms. Here, for illustration purpose, I will talk about the most studied lipid in lung biology, because it's the most abundant one in the surfactant. The surfactant is a mixture of lipids and proteins which are secreted in the lung, and their main function is to reduce the surface tension at the air-liquid interface to avoid the alveoli to collapse. This lipid, which is the most abundant one in the surfactant, is the DPPC, or phosphatidylcholine with the fatty acid chains 16-0 repeated twice. Even today, if you look for this very specific lipid on the 3 most up-to-date databases, namely Lipid Maps, Swiss Lipids, and HMDB, there is no real good association of lipid ontologies with that molecule. On Lipid Maps, there is no association with any ontology terms. On Swiss Lipid, it is associated with high-density lipoprotein particles gene ontology term. On HMDB, it's a little bit better. There are some ontology beans that have been created automatically for physiological effect, disposition, process, and role. Unfortunately, as you can see here, nothing is related to the actual function of this lipid in the lung, and it's true for a lot of lipids. If by any chance there is a program officer listening to this talk, I would love to see the creation of a lipid ontology consortium. That would definitely help advancing the understanding of lipid function in biological systems. This could be done in the same model as the gene ontology database and consortium that was created and curated by scientists. The standardized lipid names is a mine of information. Just from the name, we can reconstruct the full lipid structure. You can see that it contains the head group, the different chains of fatty acid composing the lipid. We can also calculate some extra statistics, such as the total number of carbon of the fatty acids and the total number of unsaturations. To mine the lipid names and create structural ontology beans, I therefore created a very long function full of G-subs and subs that can certainly be written in a more elegant way, but it works pretty well. I called it Lipid Minor, and it's taking a lipid name vector and convert it into an ontology database. In fact, 3 ontology databases that can be subsidized to perform creative enrichment. The first one is the lipid intact table. It's very rich. It contains the details about the lipid class, the total count of carbon, double bonds of the fatty acid chains, the list of fatty acid chains, the carbon count for each one of the individual chains, and the count of double bonds for these chains. Then there is the lipid chain table that contains the details about the chain length and the number of unsaturation. It bins the lipids into different classes: small chain, medium chain, or very long chain fatty acids. And it also does that for the number of unsaturation: saturated, monounsaturated, polyunsaturated. And finally, the lipid all chain contains the list of all the fatty acids that are associated with the lipid names. The text mining function is currently able to dissect over 48,000 known lipid species across 6 categories, 42 main classes, and 108 subclasses. For any new lipid, as long as its name follows the same nomenclature, Lipid Mini-on will be able to work with it without issues. For example, that XY lipid has the chain with the chain 00, so it's a lysolipid, and the chain 16,0 will be considered as main class XY and as subclass lyso-XY. Of course, it won't be categorized because it doesn't know what is the category of XY lipids, but it will still work for all the enrichment except this one. We have also created subsetting functions that enable for category, main class, and subclass subsetting that allows, for example, to look for the PCs with chains 16 zeros, for example. Now that you know everything about our text mining process, I will describe to you a little bit more the enrichment analysis. In query versus universe type enrichment, as illustrated with the Haribos before, we run 2 times the Lipid Minor function and we compare the ontology bins associated with each one of the 2 groups. The query, which is usually what you're interested in, versus everything that was detected, for example. We have created functions for the most popular enrichment statistics, including Fisher exact, ease, which is a modified Fisher exact test from the David team, binomial, and hypergeometric statistics. Finally, during the review process, one of the reviewers asked us to add a non-parametric test similar to the gene set enrichment analysis. So we added a Kolmorogov-Smirnov test that enabled to look at the distribution of the lipids in a ranked list. In order to ensure that anyone, even non-R expert, can do enrichment analysis, we have created a Shiny app where the data can be loaded, enrichment be performed, and results visualized and downloaded. The app was created in a tabular format with a logical order from the first to the last step. In this example, we will compare the list of lipids significantly higher in endothelial cells compared to the list of lipids quantified across the lung. This example is available on the app in the Example tab, and you can download it and play with it if you want. The software will automatically check if the lipids contained in the query are also present in the universe. We decided to perform a Fisher exact test with all the possible structural enrichment. We filtered the results to show only the lipid ontology bins with a p-value below 0.05. As you can see, in the endothelial cells, the sphingolipids were enriched, and in particular ceramides, as well as the phosphatidylserines and the lipids with very long fatty acid chains, or the PIs with the specific chain 20:4. The visualization offered in Lipid Mini-on enabled to visualize enrichment. Here you can see clearly that the sphingolipids are enriched. If we look at the main class level, we can see that the ceramide and maybe the sphingomyelins are increased in the query compared to the universe. We can also see that very long fatty acid chains are enriched on those diagrams. Finally, we can plot the percentage of fatty acid chain of one type in the query versus the universe. And here you can see a subsetting of this table where only the PI fatty acids are displayed. And you can see clearly that arachidonic acid 20:4 is enriched in the PIs. Finally, to visualize the lipid ontology terms that share multiple lipids, I have created a dynamic network using the package VisNetwork, which was very nice because it's all interactive. You can remove nodes, add new nodes, and so on. So the tool is available online at this address, and you can also access all the code source on GitHub. It's open source. And finally, it was published in Bioinformatics in April of last year. The epithelial cells are the one producing the surfactant, and using the tool, we were able to observe that the PCs were enriched in epithelial cells, in line with the surfactant production. PCs O were enriched in immune cells, and they were previously described to be rich in neutrophils, so it makes sense. We also observed that triglycerides with long-chain fatty acids were enriched in immune cells, suggesting that they might have a role in immune function. Overall, our study evidenced the collaboration of all these cells to achieve the major function of the lung, including respiration, surfactant production, and immunity. I would like to thank everyone who has participated to this work, including Kelly for the stats, Sarah Reel for the Shiny app, Jennifer Kyle for the lipidomics expertise, Matt Monroe, which helped a lot with the deployment of the app, and Charles Ansong, who is the PI of the LongMap project at PNNL. I would like to thank the sponsors as well, and you for your attention. Please don't hesitate if you have questions.